Welcome to video 5 of Genetics, the Code Broken. In this video, it's pretty heavy, but we're going to be looking at gene linkage and how that differs from the dihybrid crosses we just looked at. And we're going to be looking at how we can further understand chromosomes by mapping them out, and this relates directly to whether genes are linked or not. So when we talk about linked genes, we're talking about genes that are on the same chromosome and therefore are usually inherited together. So when we looked at that introduction to meiosis in video 4, we saw that there was 3 options. The first option, the genes were on separate chromosomes. Here, the genes are on the same chromosome and they can be linked together. And then our third option, which relates to linked genes, is whether or not there's crossing over, and that's the only way that genes that are linked can then be separated. So the further apart the genes are on the chromosome, the more likely they are to be separated by the process of crossing over. And genes that have little distance between them are rarely separated during that process of crossing over during meiosis. So here we see that we can still do a dihybrid cross, but instead of having the multiple outcomes that we saw in the other dihybrid crosses, we are again restricted to the 3x3 Punnett square. So what we saw to produce the dihybrid cross was in fact independent assortment, where the alleles are separated, different to each other, and therefore we could see the different combinations. Here we will have the dependent assortment, so therefore we are only actually getting these 4 possible gametes being produced, therefore only reducing the number of outcomes which we can see in the offspring in the F2 generation. So here, the F2 genotypes, we can see that we are taken back to the 3:1 ratio in this example, where we're produced with the dominant phenotypes of round and yellow, and there's a much lower chance of having a wrinkled green seed. So the way which we can study linked genes is by doing some test cross in breeding experiments, and this is where a heterozygote is crossed with an individual who is homozygous recessive for the trait, and it's also referred to as a back cross. And this was undertaken with the species of fruit fly known as the Drosophila melanogaster. And it's where we looked at the color of their body and the type of wing that they produced. We can see these represented with the letters B and W. So in a test cross where the alleles are not linked, we can see here that we, in our F1 gametes, we can see that there is a wide variety of the gametes which can be produced. And then we can see in the linked how we are again reduced to either the dominant or the recessive type in the gametes that are produced, and therefore the type of offspring that we can produce from that. So when we look at meiosis and linkage, crossing over in meiosis might occur, and this will happen during prophase 1 and metaphase 1, and we can see that the further, and like I said before, the further the genes are apart on the chromosome will determine whether in fact will increase their chances of crossing over and therefore recombination and increasing the genetic variability of those gametes that are produced. So here we can see here there were genes that were linked. So we can see here the dominant genes were on one parental gamete and the recessive genes are on the other parental gamete. And we know that if these genes were linked, that there would either be the dominant or the recessive, not any of those, not increasing the possibilities like we see in the dihybrid cross. But then we can see here how crossing over does in fact produce recombinant gametes that increases the genetic variability. So again, another bit of explanation about linked genes. Remember, you can access this PowerPoint on our Google Classroom. And when we're looking at linkage maps, these are diagrams that describe the locations of genes using the frequency at which these genes can be recombinated. So chromosome mapping, it provides a picture of the arrangement of an organism's genes on its chromosomes, and the frequency of recombination of selected characteristics is the basis for determining the sequence of the genes and the relative distances between them. And when we try and work out their map distance in map units, we use the following formula. So the number of recombinant offspring times 100 divided by the total number of offspring. And we can receive receive an outcome and can compare these map distances. So here we can see that when we know the position of specific genes on chromosomes, then we can plot them in a sequence and calculate the distances between them. And this is how we measure the closeness of specific genes and therefore to produce our genetic linkage maps of chromosomes. And yeah, so this has been a long process of conducting breeding experiments and then mapping all these recombinations that have been produced. Chromosome mapping would only have been possible by new technologies that have been developed, which have allowed this process to be quicker and more accurate than the breeding experiments that were originally undertaken in earlier times. So, recombinant DNA technologies produce physical maps showing the position of genes in terms of the sequence and number of the bases involved. And there's also a process known as fish mapping, where specific genes are highlighted with colors before the chromosomes are crossed, and which makes the location easier to find and more precise. Thank you for watching video 5. Make sure you tune in for video 6.